I want to thank the Bulletin and the Pathogens Project first for assembling such a spectacular group of globally diverse, institutionally diverse, and background and specialty diverse people. I think this is the most richest group of people, richest in quality that I've had a chance to work with on this topic in a long time. And also for inviting me to speak here. I want to talk about, I've titled my talk Pathogen, which you can't read, Pathogen Enhancement and Its Regulation in the COVID-19 Era. And I want to really talk about the gain of function of concern topic that was mentioned in the last session, and I'll situate that a little bit in a couple of slides of history just to remind us of how we got here with some of the highlights. And I will take a somewhat US-centric perspective where necessary, because that's what I know, and try to be as general as possible otherwise. So some of the early events I think that were crucial in helping us to all understand the potential concerns were first the mousepox recombinant experiments that raised biosecurity fears when it was enhanced with IL-4, suggesting a way to make a vaccine-resistant smallpox virus, which brought biosecurity to the front of the debate. Some laboratory accidents in SARS-CoV-1 in 2003 and 4, where individuals were infected in 3 different labs. The reconstruction of the H1N1 virus in 2001, and I'll share all these slides, so don't worry too much about writing anything down. The Purebright outbreak in 2007, which was a laboratory-associated foot and mouth disease outbreak in the UK. And then the sort of more modern history started with the controversy over the H1N1 enhancement papers that were published in 2012, and that debate continues to this day. I've somewhat grandiosely put the founding of the Cambridge Working Group on this slide, which I was one of the co-founders of, not because of trying to be grandiose, but for specifically trying to mention the importance of biosafety, which I think had been not so important in the discussion until a number of us raised it in 2014. And then the funding pause that happened in 2014 continued for several years until it was lifted in 2017 with the entrance of the P3CO guidance. And then for the period of the COVID pandemic, we had stasis in much of this discussion because people were focused appropriately on other things, and I certainly was. In 2021, the US Health and Human Services Department stated that it had subjected 3 projects to review under that guidance. Not a very large number, in my opinion, and we'll talk about that in a few minutes. And then in the last couple of years, the NSABB, which we heard about from Jerry, that he chairs, has begun to readdress these issues. So I want to make about 10 claims, 11 claims with some subparts to them. And so the first is that I think in the broad category of gain of function, meaning adding functions to organisms, almost all of it is safe, and we don't need to be talking about it. So for example, altering flu viruses to grow in eggs and cell culture for vaccines is a safe thing to do. Routine genetic engineering of other functions in microbes is a safe thing to do. And I mention this not because I think it's controversial, but because I think politics has entered that discussion. And one of the moves that we need to resist is the use of gain of function as a broad term by politicians and people with political agendas to say gain of function experiments are important, they help us make vaccines, and therefore we shouldn't regulate them. They do help us make vaccines, and that's not what anyone's proposing to regulate, to a first approximation. We can argue about the borderline, but gain of function is a very large category. The concerning experiments are not the ones that make vaccines, and they're the ones that we need to be talking about regulating. Some gain of function, which I'm going to use the term gain of function of concern. It's not a perfect term. but it's the term that I think best explains what we're talking about is risky, and specifically that which involves enhancing potential pandemic pathogens. So in 2014, Tom Inglesby and I made some calculations which we thought were on the low end with each of our assumptions, that if you multiply small risk times large potential impact, we should be concerned about causing multiple fatalities per year of enhanced biosafety level 3 work. Not that that many people are going to die each year, but that the low probability times the high potential outcome gets you to these numbers. Ron Fouchier argued in a reply to that these numbers are off by a factor of 1 billion. So we have a lot of orders of magnitude separating the two sides of the debate. I think it's worth saying that whichever end of that range you fall on, even if these numbers are off for Ron Fouchier's lab, which is certainly, as we heard this morning, even safer than certain labs in the United States, underreporting and the global variation in quality that we also heard about mean that there's opportunities for important breaches. The third thing I think is true, and Jerry also talked about this, is that the US is moving toward a pretty good definition of gain-of-function research of concern requiring enhanced review. And I say on paper because I think the devil is in the details, which I'll mention in a minute. I'm not going to read the whole thing, but I think the reasonably anticipated language is very important and a positive part of this. It means that we're not going to be able to see in advance every experiment that might generate something dangerous. But on the other hand, it will dramatically reduce risk if we focus on those things that are reasonably anticipated to create dangerous pandemic-capable pathogens. My preference would have been that this recommendation would include low pathogenicity, non-zero, but low pathogenicity, and some colleagues and I have said that in writing, but I think this is a pretty good definition. I said the devil is in the details. The biggest detail where I think the devil is in the regulation in the United States, and I can only assume it's similar in other places, though I don't know, is that we've reviewed 3 projects in the United States under this enhanced type of review over a period of 7 or 8 years. We heard this earlier from Jonas, that he has a list of the flu gain-of-function work that has been done, most of it not in the United States, but numerous projects have been performed. And among the things that were not reviewed under these for various reasons were the Wuhan bat coronavirus recombination experiments that we've heard mentioned, that were US funded, the Immunoscape selection on experiments on SARS-CoV-2, monkeypox recombination experiments at the NIAID that have been recently queried by the Republican leadership in Congress. What the purpose of those is unclear, and there is, after several months, no answer from the NIH to the oversight committee's questions. The Boston University controversy about recombination experiments with SARS-CoV-2 that came out a few months ago, and the construction of a measles virus that escapes vaccine immunity that was published in 2021. Those are some of the things that should have been reviewed if the policy was robust and applied widely. And there are various reasons, some of them perfectly reasonable given the system we have, and some of them, I think, bad judgment by those who were trying to regulate. This is one of my favorite photos. This is a soldier during World War I and the flu pandemic doing infection control. There are gaps in the US process, as in this photo. Some of them are being fixed or are potentially being fixed by changes that will happen as the US implements the recommendations of the NSABB. We review only only research that's sponsored by the Department of Health and Human Services. The review is at the time of proposal and doesn't keep up with changing aims of the research, which naturally happens in any good science lab. As I was told once by my mentor, if you know at the time if you do the research you proposed 5 years ago in your grant, you're committing scientific malpractice because you haven't learned nothing in the last 5 years. The grants are pieces of science fiction that help us to evaluate whether someone can think scientifically. They are not operating procedures. The funders' incentives appear to favor finding ways to keep proposed work out of the system, and there's extensive discretion about what goes in. And for now, until those recommendations are implemented in the US, it's only highly transmissible and highly virulent organisms that would fall in the review process. So when I say that we need a process, that is not a process to stop all gain of function of concern. The point is that all research that raises the potential of causing a pandemic should be reviewed, and then some of it should proceed because it's scientifically good, it has public health benefits, and it can be made safe enough. And I'm going to talk a little bit more in detail about how I think we can do risk-benefit weighing in an intelligent way that is not, as Rocco said, doesn't require thousands of hours of person time, and it's certainly not a full solution to the problem, but I think there are ways forward. One rule of thumb I would say is that the risks are smaller and the benefits larger when we're dealing with a current rather than a potential one, threat. And I think that was something Rocco said, although I'm not sure I totally understood your comment this morning. In particular, for example, selecting immunoscape in SARS-CoV-2 may be risky, depending on how it's done, and should be reviewed, but then probably should proceed because of the potential and actual benefits for public health. And although I'm fairly skeptical of many of these kinds of experiments, I did specifically say in writing and in other ways that when the SARS and MERS adaptation to mice was was included in the funding pause in 2014, it should be removed from that because of the public health benefits and relatively low risk. In that review process, we need transparency, and that's also been a theme in the last several sessions. And I mean transparency not in quotation marks. So the US government's transparency statement describes the reasons why we should not be transparent in the review, and specifically why the names of the people involved and the arguments pro and con for the review of proposals should be suppressed. That is not transparency, that's the opposite, obviously. I think it's kind of impressive that someone would think that they would to put that in something called a transparency statement. But you know, I don't know if I need to make an argument for this or not, but at least it might be relevant that we hold civil and criminal trials in public, and the people's names and the events, those are all made public before the person is judged guilty. So the idea that we can't put research in the public domain when it raises a potential threat to humans seems a little crazy. All right, so how are we going to weigh the benefits? I think that has been a major challenge. I would argue, and I hope to discuss with some of you afterwards if you agree, but I would argue that there are three things. First, they should be public health. If there is a risk of a pandemic from an experiment in the unlikely event of a laboratory accident, there should be public health benefits and not purely scientific benefits offsetting it, so that we're weighing lives against lives. The diagram comes from from the same piece written with Tom Inglesby, which I think is actually the most important thing I have to say. The comparison we should be doing between research that involves enhanced potential pandemic pathogens and the alternative is not, should we do this research or should we do nothing? It's not, should we do this research or should we buy a bomber? It's not even should we do this research or should we build a hospital. It's should we do this research or should we do other research that costs the same amount, and that actually is a big point because the biosecurity and biosafety is expensive, so the same amount of money could buy you a lot of other kinds of research. And should we, and what are the marginal benefits that we get from doing the risky experiment instead of the next best safe experiment? And I think that actually solves a lot of the problem. And finally, it needs to be specific and subject to scrutiny. And I'll give some examples of that in the next couple of slides. So, for example, if you look at the list of mutations identified in the H5N1 enhancement studies that were performed in the 2 laboratories in Wisconsin and the Netherlands, none of those mutations associated with transmission in those studies was novel. Every one of them had a prior publication about it. The sources are listed in this correspondence. And moreover, a number of them depended on the context. So they had the phenotype of creating transmission in one background, but not in another background. And therefore, if you tried to use simple presence or absence of those mutations to say this is a dangerous virus or this is not, you would get the wrong answer. Those are examples of a more general phenomenon, which is that throughout viruses, context matters, and one mutation changes the effect of another mutation. That's the phenomenon of epistasis. And it means that how it happens in the lab is not always informative and can, in fact, be misleading if we are using it as a proxy for what can occur in nature. So the idea that to know what to worry about in nature, we can just study it in the lab, cause it to happen in the lab, and reason from that is mistaken on several points. This diagram from an article with Alison Galvani and CIDRAP a while back tries to break down that argument, but just 3 examples of that. In a meeting we convened in Cambridge where Jesse Bloom was present and some others, we concluded that the 2009 H1N1 pandemic strain had 0 of the 3 properties which were thought to be essential for pandemic potential in 2008. 0. It didn't pass any of the 3 tests. And it soon acquired those 2 of those properties, but it didn't have them at the beginning. So our ability to generalize from several studies is limited. Other work that Jesse was also involved in was showing that the laboratory experiments on prior strains, which had shown what the effect of oseltamivir resistance in flu was, those were completely misleading when you applied them to the current strain, where in fact oseltamivir resistance went from 0% to nearly 100% in a single flu season because of an intrinsic fitness advantage that Jesse's work helped to explain mechanistically. But all the previous experiments done in other perfectly reasonable strains showed that it had a completely crippling fitness cost. So another example where trying to sort of map simply mutations to danger doesn't work. And I'll skip the last one for reasons of time. I think when we're looking at benefits, we need to be precise about the specific contribution about the risky study. And it's kind of bad manners in a scientific discussion to say that other people's claims about the importance of their science are overblown. But I'm going to do it. I hope that's not like being rude on Twitter. But this is an example that I think really typifies why it's hard to talk about these things in objective terms rather than in wishful terms. So the exceptionally interesting and scientifically triumphant, I think it was a major accomplishment to reconstruct the 1918 virus from RNA. And sometime after that was done, the authors of that study wrote a paper claiming that the that had reaped unexpected rewards. And note it says reconstruction. First they figured out the sequence, and then they reconstructed it. Figuring out the sequence is danger-free, more or less. Maybe a biosecurity risk, but certainly much less risky than actually making the virus. And they have eight or so sections in this paper where they claim the benefits of having reconstructed the virus. 7 of those sections describe things that could have been done with just the sequence or just the structure. And 2 of them describe non-public health benefits that are scientific benefits but not public health benefits. So these are not legitimate claims of what you need to do dangerous research for. They're legitimate claims of something you can learn, but there are other ways to learn it. And so again, when we're comparing the benefits, we really need to be comparing against the safer alternative, not against not doing the experiment. How should we do this weighing? I would say that pandemic risk concerns should be on a par with the kinds of other concerns that already constrain our research. Many of us deal with human subjects concerns. If you stigmatize a person in your research, there's a risk that that research will not be allowed to happen. If you risk stigmatizing a person. If you torture animals unnecessarily in your research, it will be stopped, as it should be. If you use chimpanzees, the director of the NIH proudly trumpeted that we will no longer support that work, any work on chimpanzees. If you propose a study that is better than 84% of the other studies that are being proposed, you will not get funded to do it because the pay line is 11th percentile. If you propose the best study in the world that costs $1 trillion, you will not be allowed to do it. That's not breaking your scientific freedom. That is responsible weighing of the upsides and downsides of the research. So why is gain-of-function research so hard to think about in that sort of relatively everyday context? Nobody argues that we should fund $1 trillion great idea. A lot of us argue that the 11th percentile is too low because we're in the 12th and we really wanted to get that grant. But nobody thinks we should be doing dangerous human subjects research or torturing animals. I think one of the properties is that the risk that we're talking about, and this is the kind of classic equation, that risk is likelihood times impact. But in the case of gain of function of concern, it's tiny likelihood and gigantic impact, and the product of those is really hard to think about. And probabilities that are small are hard to think about in particular. Another aspect is that the people who are put at risk are unaware, they're certainly not consenting to being put at risk, and they may be very, very far geographically from the work that's being done. Relatedly, I think the regulation and the thinking about policy on gain of function of concern collides with a number of incentives and conflicts that we have. Incentives, habits, biases, and conflicts that we have. The first is in the category of academic and scientific freedom. I think the idea that we should be allowed, someone said it this morning, that scientists think that if their intentions are good, they should be allowed to do anything, putting aside the fact that we don't get funded to do most of the good ideas we have. And relatedly, the notion that science has unpredictable and enormous benefits, which I agree with. I think a lot of that really boils down to this notion of weighing. Again, the NIH budget does not go up if we do more gain of function of concern. It probably goes down through political processes, but it certainly doesn't go up. And therefore, if you're talking about a fixed budget, it's not a question of doing no research or doing research. It's a question of doing better, safer research or more dangerous research. Sometimes the more dangerous research is worth it, but let's make the case. Another point that I had not thought about, but my colleagues at philosophers at Rutgers pointed out is this very strong feeling that we have that we should do something. That if we have a threat, if we have a potential pandemic pathogen, we should do something to study it, no matter whether that's a good idea or a bad idea. And if you haven't read this paper called Cure Me Even If It Kills Me, Preferences for Invasive Cancer Treatment, it's quite a fascinating paper in medical decision making about how patients think about cancer treatment. Another related point is that we are unfamiliar with and unused to regulating research risks to non-participants. All of the machinery that was built up after the Second World War to regulate risks to humans from research was about human research participants, for very good reason, because that is a very risky kind of research. But this graphic from our commentary in PNAS about the idea of non-participant risk points out that when there's a transaction between, say, a clinical trial involving treatment of an infection between an experimenter and a participant, the non-participants may have some risk as a result of that. And we surveyed in a couple of different areas the literature on whether there's any obligation to protect non-participants, and it's a very murky frontier as we described in this paper in Clinical Trials. I think there is also obviously the funder institutional and other incentives to do and fund and publish research in synergy with entrenched positions and false entanglement with other topics. I don't know where that bird came from, but it might have something to do with that. And then finally, a tendency to underestimate small probabilities, which is a psychological bias that's been well documented and I made allusion to a minute ago. And actually strong incentives in people who are trying to maximize their career potential or their other outcomes to take inadequate care to prevent small risks. And that was something we also wrote about in thinking about why we don't adequately protect against very low probability but high impact events. I think we have dealt with the topic of gain of function of concern in the setting of enhancing flu viruses. And I think David Relman and I wrote a piece in 2015, that predicted that it would get harder, and we didn't know exactly how. But I think some of the ways that we thought it would get harder have come to pass. The early experiments dealt with known and named pathogens for which we knew a fair amount about them. They involved expensive government-funded studies in labs that were known and public. And they involved a limited profit motive. It was mostly a matter of public goods versus public goods. I think that's going to start to change, and some of those things have changed. For example, the possibility that viral prospecting and viral discovery experiments could lead to starting a pandemic. Whatever you think of the COVID origins question has now come onto the table and raises the question of how to deal with that, which I don't know the answer to. I think we've heard a little bit about the role of journals, and of scientific journals, and I want to reinforce that thought. This is Daniel Bernoulli, who was the first mathematical epidemiologist by many people's accounts, also a Swiss person, so I wanted to put him on my slide. And he published in this journal in Paris. But a lot of us in more modern dress have other things on our mind, and some of them are more prominent than others. But it's clear that the ability to publish in a prestigious place is a major motivator for scientists. And that gives the journals the ability to have influence. We can't publish anywhere respectable if we haven't followed human subjects guidelines and animal guidelines, irrespective of who funds us. That's not the funders, that's the journals. More and more, if we don't make our code available or our data available, we can't publish. So the journals have figured out ways to incentivize behavior by scientists. How do we do that? I think one way to do it would be for journals to insist on certification that proper biosafety and security have been certified. And I don't think that would be the perfect answer, but I think it would help. And it would have a global effect irrespective of who funds, and it would help to educate people about the issue. We've heard about the importance of internationalism and the value of a conference like this in trying to incentivize it. I think we also need to keep in mind that one country at a time making some progress will help. It will set a global example if the large research funders make progress, and at the same time we need global thinking to help us solve these hard problems. The Washington Post had an article last week or the week before about a Thai scientist who had said to the US government, "Thank you for your viral discovery funding. I think this is dangerous and actually not really helping public health, and I'm going to give it back to you." That voice is not exactly well, that voice is Thailand educating the United States, in my view. Of course, the people affected are everywhere, and so that makes it a global problem. Funding crosses borders, if not the viruses. And nonetheless, as much as globalism matters, each time we make some progress in one country, it will help. I got a little lesson in French from my friends when I was thinking about this slide. Apparently in French this is not true, but in English a canard, I mean I know what a canard is in France, but it's not, and in French, but apparently it does not mean a falsehood that is repeated for effect. But in English that's what it means, as most of you know. So we have to counter some of the canards. And I appreciated Zeynep's doing so last night for her whole talk. There was a lot of canard hunting. Some of those canards are responses, reasoned responses to some of the canards that are most important are, first of all, gain-of-function research of concern is not producing vaccines or other countermeasures. Not to say it never can, but it is not right now. And when prominent people go on television and say gain-of-function major flu vaccine, that is not helping the discussion. Regulating this research is no more of a threat to academic and scientific freedom, I think, than human and animal regulation, open science, and so forth. Third, I think really importantly is that harm reduction works. We are not going to get to perfection, but making better policy and better mechanisms to implement that policy will make us safer, and we should try to be incrementally beneficial rather than waiting for the perfect thing. Relatedly, I think there's been a suggestion that if we regulate in one country, it will just push the research to elsewhere. I think that's not, it might have that effect in the very short term, but I think if the research is limited in certain countries and and builds a culture in which that is not, in which you think twice and ask the question, does this need to be done in the most dangerous way, or are there ways to make it get the same public health benefits more safely, that will filter to the whole world, because nobody, as we've heard, nobody is anxious to have an accident. It's simply a question of what kind of research people choose to do. And finally, I think with all the interest of the last session, I want to just emphasize that the wisdom of whether we regulate gain of function of concern and how we do it has almost nothing to do with the COVID origin question. So in conclusion, I think we are at the COVID-19 has brought us new salience to this notion that accidental pandemics would be bad and something we want to avoid. I think this meeting has done a really good job of assembling a really concentrating a very diffuse solution of people who are in the reasonable middle. I don't know everyone's views in the room, but I think there are more people in this room who have moderate views and are interested in trying to work together to understand the competing views and make sensible rules than almost any other group I've been a part of. Having said that, I think that there are still a lot of research projects happening that have little or no public health benefit and create real risk of causing outbreaks or a pandemic. But we have to preserve a way to do the limited subset that is both risky and incredibly valuable. I think there are ways to close loopholes and do the risk-benefit weighing that are not overly burdensome. I think if we really adhere to the concept of comparing research that creates potential pandemic pathogens or enhances them against the next alternative that is not as risky, we will solve many of the benefit questions. I think we need a lot more thought about the viral discovery question, and do we need limits on it? Is it dangerous, and what are the short-term and long-term benefits of viral prospecting, and where should we do it and where should we not? And clearly now is the time to act. We heard that it's been 10 years of not following up on recommendations. We have the salience of the pandemic, and if we're not going to do it now, we're not going to do it. So I'll stop there.